So at TTLC 2022, I'm going to speak about small cell lung cancer, which is the most aggressive form of lung cancer, and it's in a critical need for novel therapeutic opportunities. And I spoke about an epigenetic modifier, LSD1, and its role as a therapeutic target in small cell lung cancer. So as I was saying, small cell lung cancer is really the most aggressive form of lung cancer, and it accounts for about 15% of lung cancer diagnosis. It is characterized by very rapid development of drug resistance, and the frontline therapy for small cell lung cancer, which is currently chemotherapy and immunotherapy, has very modest benefit. So there really is a very critical need for novel therapies and novel approaches for small cell lung cancer. Now, one of the characteristic features of this cancer type and why immunotherapy does not work as well as it should is that it has a downregulation or a suppression of MHC class 1, which is a protein that's needed for antigen presentation and also T-cell priming. So what we know from previous studies is that in small cell lung cancer, MHC class 1 expression is low, and it is transcriptionally repressed by epigenetic modifying complex. So we have seen in the past studies that PRC2 complex plays a role, and treatment with epigenetic modifier, targeting an epigenetic modifier drug, EZH2, has a benefit in small cell lung cancer. So in this study, what we looked into was a second epigenetic modifying target, which is LSD1, also known as lysine-specific demethylase 1. And it has its role in like cell proliferation and differentiation. And previous studies have shown that LSD1 is upregulated in several liquid tumors, but also in some solid tumors, and it has been linked to carcinogenesis. And in this study, what we have shown is that targeting LSD1 upregulates MHC class 1 in small cell lung cancer, and also it augments or potentiates the anti-tumor immune response of anti-PD-L1 in small cell lung cancer. We have done in vitro studies, and we have done studies in mouse model, and we have shown that either treatment with an LSD1 targeting drug, either as a single agent or in combination, is very effective for small cell lung cancer. So I think from our presentation, the key take-home messages are that LSD1 inhibition, it decreases the viability of small cell lung cancer cell lines, which are usually quite aggressive and proliferative. And small cell lung and LSD1 inhibition increases the expression of MHC class 1 genes like HLA A, B, and C in small cell lung cancer in vitro and in vivo models. And LSD1 inhibition increases also genes that are involved in antigen processing and antigen presentation, and also cytokines, which leads to a better anti-tumor response. And finally, I think, which is the most interesting part of our study, is that LSD1 inhibition augments the anti-tumor immune response of PD-L1 in small cell lung cancer, very aggressive immunocompetent mouse models. So I think this would be an interesting study, an interesting clinical trial to follow up and see how this drug actually behaves in patients.